Welcome dear students to another class, Biology by Dr. Shabana Khan, and this is our series of lectures on inheritance for O levels. And this video lecture will be on test crosses. Okay, so let's see. So the topic is a test cross. Now, what is a test cross, or what is the objective? So you're trying to test for something, right? So what are you trying to find out? So the aim of a test cross is to find the unknown. So underline the word unknown genotype of an organism. All right, to find out the unknown genotype of an organism. Right, now let's take the example of mice. All right, and mice, and the gene we're concerned with is going to be their coat color. All right, so the gene is going to be of coat color. Now in mice, the coats can be, for example, the species that we're studying, they can either be brown coat color or they can be white coat color, right? And then we got to know that brown is more common, so we say that is the dominant allele, and whereas white is less common, so we say that is the recessive allele. Now, how do we write it down? So alleles are written as two letters. For example, allele, if it is brown in this case, that is the dominant trait. So we will use the first letter of the word brown and apply it here because it's a dominant one. So we'll use the capital letter B. Now the other one is the recessive one, that is this less common, and we have said that color is actually white. So this is white. Now, white coat color is the recessive allele. So that means this will not be present. This will not be apparent in the phenotype if we have the dominant. That's why it's called the recessive. Now, what letter do I use for that? Do I use W? No. It becomes too confusing. So what will I do? I'll take the lower case of the same dominant letter, capital B and capital B and small b, right? Now, what will be the genotypes and the phenotypes? What are the possible combinations of alleles in this mice, right? That's what you mean by the genotype. Now, there are three possible genotypes. What could they be? Either both of them are capital Bs, or one is a capital B, the other one is a small b, that means dominant recessive. Or the third option could be that both of them are going to be recessive and recessive. Now, capital B, both of these will present as a brown coat color, and this one will also represent as a brown coat color, whereas this one, we are 100% sure, because this is white and this is white, will only represent as white. Yeah. So now the confusion starts here, that if I see the phenotype or the coat color as being brown, then what the question would be, that what will be the genotype? Because I can't see the genes, I can't see the alleles, they will be present in the DNA. So the brown phenotype or the brown physical form would either be this or this. This is what? Homozygous, dominant, and this is the heterozygous form, right? It could either be this and this, but we don't know about this. We can see this, we can't analyze this. So what do we know? How to get this? This is the, what we say, unknown hidden genes inside, and this is unknown. So how do I find it out? I'm going to do a process known as a test cross. All right, so that is the basic background of this question. Now let's see. Now, so for example, let's start again. Now the question is that there is a mice and it is showing the dominant allele or dominant phenotype, and that is the brown color. But the genotype is unknown. So the genotype is unknown even though it's showing the dominant trait. So what does that mean? That this could either be, let's call this Mr. X. Mr. X, we don't know what this is. Now this could either be capital B and capital B, that is homozygous, a pure breed, or it could be, that means or. So we don't know, right? This could be a heterozygous form. How do I get to find out this? Is there a process? Is there a way by which we can find out? Yes, we can do a test cross on this, right? Now let's see. So what are the steps, you might say? Now the steps involved in for a test cross would be, number one, what do? We're going to cross this organism X, which has the unknown genotype. We're going to cross this with a white one. All right, so we say cross. Cross means we're going to breed this. Cross the unknown one, X1. I just can put a question mark here. So that is having the unknown genotype. Or you can write it down. Unknown genotype with a homozygous recessive one for the same allele and for the same gene, and that is a white mouse. All right, so I'm crossing the brown one with a white one. All right, so we cross this, and then what do? Step number 2 is that we will observe the phenotype ratio in the offsprings. All right, so and then after crossing the brown one with the white one and the homozygous recessive one, we're going to observe what will be the ratio in the offsprings. Now, theoretically, we know, so theoretically, we already have worked this out. This is, for example, organism X, and if we cross this with what? Small b and small b, or the homozygous recessive one that is here, all right, and or if this we cross with this, all right, and this is the X. So supposedly, if this was a homozygous dominant or a pure breed, or if this was a heterozygous, then what would be the outcome in both of these cases? Let's draw a Punnett square. Now, in this case, if we do this, what will happen is that the genes, the alleles of one, the gametes of one parent would come over here. This is one organism, one of the mice, and then the other one will be placed over here. And then we can fill up the test cross, the Punnett square, capital B and small b, capital B and small b, and capital B, small b. Now, wherever you have the capital B, that is the dominant trait, the dominant trait would be there. That means all of them would have the brown coat color, brown coat color, brown coat color. So what do we know? What is the phenotype ratio? Will be that 100%, right? So they will be, the offsprings would be 100% be brown, or 100% will be showing the dominant trait, right? Now, what about in this one? If we draw a Punnett square, we can easily make out that, just remove this line from here, right? And that would be that these two will split up, and we draw a Punnett square. So for this, the gametes would have this, and then they would have this. Now, when we fill it up, the Punnett square, it looks like this. All right, now as you can see, both of these have capital B, so they will have brown and brown, whereas there is no capital B here, so this will give us the recessive trait, right? What will that be? White. So the result comes out that the phenotype ratio is brown against white, or dominant against recessive, is 2:2, or simplified as 1:1, or we can say 50% chance of either. 50% chance of either. So this is the theoretical background. We already know this. Now, the conclusion that we can draw, the third step would be that we have to conclude from this test cross, and that is that if all offsprings, right, are what we say showing the dominant trait, like we said 100% are there, 100% are showing the brown or the dominant trait, then the unknown genotype, right, in this case would be capital B, capital B. In other words, it is a pure breed. and this will be a homozygous dominant organism. But if in this case, if some of the offsprings are going to show what? The recessive traits are showing dominant and some showing recessive, or if there's a ratio of 1:1, you can bring that in. So that means that the unknown genotype is actually a heterozygous form, or we can say it will be the heterozygous form. So what do we conclude from this? A test cross is done to find out the unknown genotype of an organism showing the dominant trait. In this case, it was the brown color. What do? We're going to cross this organism with the one which is showing the obvious recessive trait, and that will be the one having the homozygous recessive form, right? And theoretically, we know that if it was a pure breed, then all of the offsprings in the F1 generation will show the dominant trait. There's no absolute zero chance of it coming out as a recessive. But if it was a heterozygous or a hybrid, then some of the offsprings might come out with the recessive phenotype, and that will be the conclusion of this. Okay, and that is all about the test cross. Thank you.